Hello, my name is Carlos Rafael, and I'm in Bio 340 class with Professor Palmier. And today I will be talking about two different techniques that we can use to identify the presence of a DNA or an RNA in a gene. I will be talking about the nucleosome protection assay and the northern blotting. First one I want to talk about is NPA, or nucleosome protection assay. So it is a test to see if genes are ready to be transcribed and expressed or not. One thing I want to mention about this test is we have to make sure that histones are degraded. In other words, the presence of histones protects DNA from degradation by DNA nuclease or DNase 1, which is the condensed chromatin. So we have to make sure that DNA is susceptible to degradation by DNA nuclease or DNase 1, which means that the histones should not be bonded to the DNA. In another way, it is the decondensed chromatin. Some modifications that could help in degradation are the acetylation of lysine or phosphorylation of serine. Another modifications that wouldn't really help in degradation are the methylation of lysine or just having normal lysine or non-phosphorylated serine. In general, NPA is a yes or no question if a gene is there or not. The process of NPA starts with DNA isolation, or the DNA has to be cut into small fragments with restriction enzymes. After that, the DNA will get separated by size using gel electrophoresis, which is basically a large to the top and small to the bottom. Right after that, we will do a southern blotting, which will be performed by putting the DNA in a membrane of alkylation solution. Then it will be exposed to a probe. A probe is basically a small complementary sequence of DNA and RNA together, which will base pair with a gene of interest. Last step will be X-ray film, which will be done to see if the gene of interest is there or not. Now we get to the northern blotting. It is basically a test that looks at the RNA levels in the gene and to see if a gene is transcribed. It basically measures the increase in mRNA, which basically means the increase in transcriptions. In this technique, genes has to be loosely bound to histones again in order to express the gene, as we said before, an NPA. Also in the test, the degraded gene has less darker color than the saturated gene that has an increase in the transcription. And we will talk about this later. The northern process starts with the same kind of steps in NPA, the isolation of RNA to ensure strands are unfolded and they are not bonded to each other. Right after that, gel electrophoresis will be done to separate RNA molecules. A southern blotting will be performed by exposing RNA to a probe, which is a small complementary sequence of DNA and RNA together, which will, again, base pair with the specific RNA. Finally, X-ray film will be done to see the RNA of interest is present or not. So for my experiment, the gene that I am going to use, it's called the BRCA, which is a gene where a mutation can happen that causes breast cancer. So for us to determine if the gene is there or not, we will take it both ways, from the NPA perspective and from the northern blotting perspective. So from the NBA, we are looking at if the gene is there or not. So we'll basically get chopped up and degraded by DNase 1 and will be shown on the test, as I included in my NBA process earlier. On the other hand, for northern plotting, we are measuring the RNA levels in the gene, and we can use northern plotting to look for the specific RNA and to measure the increase or the decrease in transcription, as I said. One more thing, just to be clear, that the RNA molecules will show a dark or less dark according to the amount of transcription we have. Now I will be demonstrating how we draw the NPA and northern plotting. So I will be starting with NBA. So first, let's draw it all in here. So I will be comparing two things, that the BRCA bonded to a histone, as I said, this is not degraded, and the degraded BRCA. So I'll put here a BRCA with histone, and here the degraded BRCA. And again, we said what causes the degradation is DNase 1. And that will be in micrograms per milliliter. So again, this is, we will be doing this comparing 48 hours to 96 hours. So let me just put this in here. So at first, the BRCA that's bonded to a histone will show just a dark band right here, as we see, because histone is protecting the DNA from degradation by DNase, as I said. On the other side, with the degraded BRCA, we will see it looks like this. It's dotted, means it is chopped. Basically, the degraded BRCA will be chopped up by a DNase 1. And from this test, we can conclude that there is an actual presence of the gene, since it was degraded, and this is how we see it in here. Now I'll be doing the northern plotting. So let's draw this one more time. For this one, I will be comparing the untreated DNA with the 48-hour one. And the 96 hours one, after 96 hours. So the untreated one, we will not see any bands for it, since there's actually nothing bonded to it. But for the 48-hour, we can see a circle that's light, which means that there is transcription, but not enough transcription to be shown. Comparing this to the 96 hours one, we will see that it is darker circle, which means that this shows that there is the RNA levels, or there is transcription occurring, and it is increasing. Just to reiterate what I said, the test shows the presence of the specific RNA molecule. As the time passes, transcription is increasing, which means that the mRNA is increasing, and that what is shown in dark color at 96 hour. just final word, transcription is not strongly present or increased at the 48-hour. That's why it is shown in a less dark color. Again, thank you for listening to me.